My name's Alison Stewart, and today I'm going to give you a talk about the research that I've done over my academic career, focused on growing a greener future for New Zealand's primary industries. And in particular, I'd like to talk about the work that I've done in New Zealand horticulture. And I think we all recognise that the fruit, vegetables and grain crops that we grow in New Zealand are high quality and very safe, and they've been produced using very resilient and sustainable growing systems. But I think we also have to recognise that the temperate climate that we have in New Zealand provide conditions where weeds, pests and diseases can thrive. And so in order for us to be able to produce high quality crops, we have to have very rigorous and stringent crop protection practices. And over the last probably 50 years, we have tended to rely very heavily on agrochemicals to be able to control those pests and diseases. Nowadays, though, we find that the consumer and I guess the agrochemicals themselves are preventing us from being able to utilise them in a very effective way. And so we now have a situation where there is a driver to move away from heavy agrochemical use because, as I said, some of the chemicals are no longer working as well as they used to because of resistance building up in the pest population, also because of increased residue tolerances, and also because of particularly increasing consumer demand for low pesticide use. And what this has meant is that there has been a lot of work over the last 20 years looking for alternatives, and particularly biological-based solutions. And so what we find now is that there are new categories of crop protection technologies coming into play. And so we have biopesticides to be able to control pests and diseases, biostimulants to help us stimulate plant growth and tolerate environmental stresses. And we also have a range of biofertilizers that can replace some of the synthetic fertilizers. And this is the area that I've spent my academic career focused on, and in particular the development of novel biopesticides. And when we're talking about biopesticides, I'm talking specifically about beneficial microorganisms. So these are naturally occurring fungi and bacteria that can inhibit the development of pests and diseases. So when I first started out, I went around the countryside, particularly looking for very diverse ecological niches, and I collected a large number of strains of microorganisms, and I took them back to the research lab and I identified and characterized them. And during that period of time, what I noticed was that there was one particular group of fungi, beneficial fungi, that were very common and were very bioactive. And this is a fungus called trichoderma. And so I decided to try and focus on trichoderma, and I collected thousands of strains of trichoderma into a culture collection, which we called a trichobank. And you can see in this photograph on the left-hand side, that's the trichoderma fungus growing on an agar plate, and then the two on the right hand side show you what it looks like under the microscope, where you can see all of these filaments and you can see that the fungus can produce lots of spores. So once we decided to focus on trichoderma, we then developed a range of bioassays to try and identify the types of bioactivity that the fungus could exhibit. And we looked for its ability to grow on plant tissue. We looked for its ability to parasitize other organisms. We looked for its ability to be able to produce antibiotic compounds. And we looked for its ability to be able to promote plant growth. And on the basis of all of those bioassays, we selected strains that looked as though they had potential to be able to be commercialized. Now, obviously, all that work was done in a laboratory, so we had to take those strains out into the field environment and see how they performed. And it's very difficult to be able to follow microorganisms out in the field. And so we had to develop a wide range of novel detection technologies. So we developed culture media that only our fungus could grow on. We developed molecular markers that would identify our strain, and we also developed techniques to fluorescently label our fungi so we could visualize them in situ. And this allowed us to do a large number of different population ecology studies, and which, we could say, where does our organism grow and proliferate? What environmental conditions help it or hinder it? How compatible is it with all of the other control measures that the farmer is applying to his crop? And it allowed us to select strains that were able to perform well in a field environment. The other thing that we did was we looked to see what genes were involved in the production of that bioactivity, whether it was a gene for mycoparasitism or a gene to express a particular antibiotic. We also played around with protoplast fusion, which is basically taking cells without any walls and fusing them together to see if you can introduce multiple attributes into the single strain. And this was very challenging work, and it involved 20 years of development work being conducted mainly by very able postgraduate students and postdocs and technicians. But what it did allow us to do was develop strains that were robust in a field environment. So they might have a very broad temperature tolerance, or we might have been able to develop fungicide tolerance so that the strain could be applied with a complementary fungicide. So obviously that's research work. We developed a business partnership with a company in Christchurch called Agrim Technologies, and we work together to be able to scale up and commercialize some of those strains. And to do that, you have to learn how to produce it at scale. And we grew it up by solid state fermentation processes. We also had to learn to develop novel formulations so that it was stable and it could survive in the field. And we also had to work out what was the best application strategy for each of the different strains for the different targets that we were focused on. And so at the end of all of that, we did commercialize a number of products, and there's just a few illustrated here. For example, Tenet was a biofungicide that we developed to control a disease in onions, and Sentinel was a biofungicide that we developed to control botrytis disease on a whole range of fruit and vegetable crops. So that was during my academic career, and I decided I then wanted a change. And so I went to work for a large biopesticide company in California. And that was where I really understood what the challenges were in being able to take products to market. And during that 3 years that I was overseas, I was also involved in the development of a number of other commercial products. And when I returned to New Zealand, I decided that whilst it was challenging to develop commercial products, I actually felt that developing the product was probably the easy bit, and getting the growers to adopt the technology was probably going to be the hardest part. And so for the last 10 years, I've been spending my time involved with growers trying to encourage the adoption and uptake of biopesticides. And that is no mean feat. It involves a lot of developing grower resources, manuals, information booklets, because many growers have never experienced using biopesticides. They don't really know what they are, and they don't know how to use them and integrate them into their farm system. So a lot of extension work, a lot of seminars, a lot of conferences, webinars, question and answer sessions, a lot of field trials, a lot of demonstrations to show growers that there are other ways in which they can control pests and diseases in a more sustainable way. And that's what I'm going to continue on doing in my semi-retirement. So I guess a big question that people ask is, what is the future of biologicals? I've spent 40 years of my life developing novel bio-based products, as have many other researchers, both in New Zealand and overseas. Well, I think I can confidently say that there is going to be a promising future for biologicals. They won't replace chemicals, but they will become an integral part of an integrated disease and pest management practice. And so I envisage that growers will use tolerant cultivars, and they will mix and match biologicals and synthetic chemicals to help them control these invasive pests and diseases. And what I would really like to see over the next 10 years is that New Zealand transitions away from a chemically intensive production system to one that is more sustainable, where the biologicals and chemicals are used in a judicious way, with the bio-based products as the base of the programme and the chemicals only being used when they're absolutely necessary. And I would like to think that New Zealand is already progressing towards that vision. And I think I've been very fortunate in my career to be able to contribute to that transition and to hopefully see New Zealand grow a greener future. Thank you.